I only have a minute, so let's talk about the leading versus lagging strand.
DNA is known as antiparallel. That means that one strand of DNA is going to go in one direction, while the other strand is going to go in the opposite direction. If we look at this strand of DNA, we can see that this side ends in a phosphate group, while this end ends in the sugar group. If we look on the opposite side here, we can see the top ends in a sugar, while the bottom ends in a phosphate group. This arrangement of the molecules is known as antiparallel. DNA polymerase is the enzyme that's going to copy DNA. It makes DNA in a 5' to 3' direction. This means the DNA polymerase is going to run along the original strand of DNA from the 3 to the 5' direction. On this bottom strand, the DNA is going to be made in a continuous path. That's known as the leading strand. The other strand is known as the lagging strand. That's because it has to go in the opposite direction. That's because DNA polymerase can only run in a 5' to 3' direction. One way to remember this is that on the leading strand, DNA polymerase will go towards the replication fork. On the lagging strand, the DNA polymerase will always go away from it.